All right, we're starting a new series of lectures now in a completely new area for us. We've been leading our way into that by talking about linkage. Now we want to talk about the chromosomes themselves. And certainly we've done that when we talk about chromatin structure early on. We kind of described how chromosomes are put together with nucleosomes, and we talked about the condensation process. Now what we want to look at is once you get those chromosomes where you can see them, how do you study them and what do you get from studying them? Okay. So as I said, this is one of the major areas, 4 areas that we've broken genetics down into. We looked briefly for what we would call molecular genetics. We'll come back to that big area in just a little while, but we've been off for a while now in this area that we would call transmission genetics. And transmission genetics here, and kind of using my little illustrations. Art history was probably was my favorite class, and when I was in undergraduate, and so I've carried that with me. I love to go to museums and things. But anyway, using those little examples here, we said that what Mendel brought to our understanding of genetics was much like the great Dutch masters. They brought this idea of shadowing, and they brought perspective where you can look forward, and it really brought dimension to what we were seeing. Mendel did the same thing. He brought dimension and understanding by his principles so that we could work from those. And what we're going to do with cytology is really come along and sort of expand on that. We're going to take these basic Mendelian ideas that are underneath things, and we're taking this whole concept here of perspective and maybe shadowing, and much like the Hudson River school did when they came to do these big landscapes, and they started doing great depth and bringing in this dimensions. They just took the basic ideas and sort of moved them forward into a larger area. Same thing here. We're gonna take these concepts of transmission genetics and now really go look at the big picture that's on the outside. After that, we'll return to molecular, and we'll actually go down here and turn the rocks over, see what's under each one of those. But for now, we're looking at the big picture. All right, so just a couple of definitions. Cytology is the study of chromosomes. Ology is the study of, right? We see that. And cyto in this case means chromosomes. We're going to concentrate on a sort of an area of this called cytogenetics. And this is the study of the number and structure of chromosomes in plants, animals, humans, whatever. Okay? Now, we're actually going to concentrate for the most part on chromosomal anomalies, looking at variations from the normal chromosome set. So kind of understand how that goes, because that's really where some of the interesting parts are. But in reality, cytology is far beyond that. When we could first look at chromosomes and band them and sort of find them really well, and particularly into the 1960s and 70s, it was a key technique, but long before we could sequence DNA effectively. It was a key technique for things like looking at species differences and defining, well, this has a completely different chromosome set than this one. They probably can't hybridize, so they're probably different species. Also for looking at human diseases and all those sorts of things, it's very important. So it's a big, broad area, and we're going to concentrate on sort of looking at the number and structure of these chromosomes. Now, what I need to do in this lecture is work through a few terms so that we're all familiar with some basic ideas. And have a common vocabulary. And then we'll finish up by talking about some of the mechanisms that might actually lead to some of these chromosome anomalies. After that, we'll have a whole series of lectures looking at some of these anomalies. What could they be? What would depart us from sort of the standard chromosome set? Okay, so first some definitions. The first definition we want to talk about is a karyotype. A karyotype is basically a picture of our chromosomes, generally taken in late prophase, and generally taken in a situation where they have been modified. The cell has been modified by some type of mitotic inhibitor. So this is usually during mitosis. We don't want to form the synapse, although that's when the tetrad is formed, that is an interesting time to look at cytology, but for most karyotypes, we simply do it in a mitotic phase because A, we can get cells in mitotic phase a lot easier than in meiosis, and we can also arrest them. Usually there's some type of mitotic inhibitor. Coltracine is a classic one used for many, many years. What it does is interrupt the formation of the spindle fiber mechanism. So you don't ever really move out of prophase into true metaphase because the system's not there. And it's one of a number of different inhibitors. It's actually developed originally for treatment of gout, but it turns out that it breaks up these things. So what happens is if you apply colchicine to a cell culture or to tissue, and you have cells that are going through the process of replication and then to mitosis, they'll arrest in late prophase, and you can take a picture of them like we did here. Okay? Once you have that picture, then you have to put the chromosomes together. Remember, we have homologs, right? So we have one from mom, one from dad. By the time, of course, you get into prophase, they've each replicated. So we have dyads. We put those dyads up. So if you line them up into some figure, usually from largest to smallest, okay, and they're lined up with multiple ways. Now, just to kind of, bring you perspective of where we are, each one of these is a dyad, so that's a homolog of that. This is chromosome number 5, pair number 5. This happens to be humans. This is a male human. Chromosome number 5, okay? This is one homolog. Maybe that came from mom. Maybe this homolog came from dad. Each side's been replicated. So really what you're looking at is, for this, is this right here. And there's a little centromere right there. So one of the ways that we arrange them tends to be group them by size. We tend to put in the centromeres kind of where they belong. And then we also look for these things called satellites. You particularly can see that right here, these little areas where you've got a small amount of DNA and then a kind of a large area that takes up a lot of stain on the outside. Those are known as satellite systems. So largest to smallest. This ordering of our chromosomes going from the karyotype to this is called an ideogram. Okay, an ideogram is the way that we would standardize the way we would illustrate which chromosomes we have. We pick them out and place them this way. So now you'll see in a lot of places this is called a karyotype. That's not really what a karyotype is. Karyotype technically is the picture that you take of the late prophase chromosomes. An ideogram is then taking those and actually structuring them out. Now, in the old days, the way we used to do this is we take that picture, or literally take a physical picture, and we would take a pair of scissors and we'd cut out each one of these and then sort of tape them together based on that. And they're homologs, so you can compare them by, they would have the same length. You try to find ones where the centromere is in the same location, and of course we would know then also that the loci would be the same, but you can't see the loci here, but that's how, with a homolog, that's our definition from earlier lectures. This is form an ideogram. And nowadays you don't do that in quite that way. It's now all done automated. You have specialized stains that can come along and stain particular chromosomes. And then you can match those chromosomes up based on the staining pattern as well as the centromeres and those sorts of things, so that you can end up with a fairly simple way for computers to actually go through and to organize this themselves. So you don't really hand cut them out anymore, but you would sort of do this, okay? All right, now, the question might be, in today's world with the ability to, do DNA sequencing overnight and to really look at our genome, why in the world would we be interested in cytology? And why were we interested in it to start with in some ways? Well, probably it applies to all plants and animals, but probably to bring it home the best, we should talk about maybe humans. All right, well, so we'll use a lot of human models here when we can. Sometimes we can't because things don't really occur that way in humans, but when we can, we're going to mostly use human models and examples for ours, okay? So just to give you some ideas, think about humans. Somewhere around 50%, and some estimates all the way up to 75% of fertilized eggs never actually implant. So you go through the process of reproduction, you go through fertilization, but those eggs never implant, okay? When they do implant, so you have a known pregnancy, if you will, the estimates, and these numbers that I'm going to give you will vary. So if you go around the web, you might see slightly different numbers, but it doesn't matter. I'm kind of making the point here. Of those that do implant, so vast majority don't even implant. Of those that do implant, so that they're there long enough to have a known pregnancy, right? So within, say, 12 weeks or so, first trimester. The estimates vary, but somewhere between 15 to 30% to 20%, all the way up to 31% of these known pregnancies will end in what are known as spontaneous abortions, known as miscarriages, right? And of course, probably everyone here has either a family member or knows an acquaintance, or maybe even you, has been through a spontaneous abortion. These miscarriages, they're quite common. They're certainly common for many reasons, right? You can have first pregnancies have a higher incidence, health of the mother, uterine conditions, all sorts of things, stress, all sorts of things can lead to these miscarriages. And they're fairly common, okay? And what we're interested in is the cytological relationship here. So of these spontaneous abortions, upwards of 50% of them, when you look at the abortices, are actually due to chromosomal anomalies. So basically, vertebrate systems, particularly mammalian systems at least, humans, a good example of that, really the imbalance that occurs due to having not the right number of chromosomes causes all sorts of developmental issues and timing and things to go on. The systems recognize that and they simply terminate the system. They're just not viable individuals. So very often when you have a chromosomal anomaly, you're going to actually lose that. In fact, you look at some statistics, somewhere around 98% of chromosomal anomalies are actually aborted. Okay, how do we know this? Because we look at abortices and we can see of stillborns and things. So they're aborted, okay, spontaneously aborted, which means that we lose them within about the first 20 weeks of pregnancy. Once you get past that 20-week mark, once you get past the 12-week mark, which is where most of the spontaneous abortions occur, once you go past that, toward 20 weeks, you have a much greater chance that you'll make it. But once after 20 weeks, it's called a stillbirth, right? Because they're much further along in development, but they're not able to function as living humans when they become air breathing. So what we see here is we see somewhere around 98% chance that you're going to abort that, spontaneously abort, miscarriage, that anomaly. So very few of them actually make it to past 20 weeks. About 1% make it stillborn, so they make it, but they're just not functional at birth, parturition, and only about 0.6%, and that number can go up and down, but about 0.6%, less than 1% for sure, will actually make it to live birth. Okay. Now, to put this in some perspective, the estimates right now are somewhere around 25,000 to 30,000 children born each year in the United States with a chromosomal anomaly. Now, many of these are very minor anomalies and all that, but some are very severe. But still, we have some estimate of about up to 30,000, and some estimates take it as high as 40,000 children born each year with a chromosomal anomaly. Worldwide, that's somewhere around 1 in 200 births. Sometimes you see 1 in 150, but it upwards, I think more recent estimates put it about 1 in 200 births worldwide involve some type of chromosomal anomaly. Again, some are very minor, some are very problematic. Now, think about that in reverse. If the live births, the born here, are represent about 0.6% of what you actually have, if we backtrack the numbers here, we're talking about millions of conceptions that actually involves chromosomal anomalies. So even to this day, no matter what kind of health you provide for the mother and the womb conditions and all that, still just mechanically getting that fertilization event to occur, getting all those chromosomes over there correctly, going through that whole process, and then not losing any through meiosis or losing any as we fuse those gametes together. All of that is a place that anomalies can occur. And in fact, they do. And they apparently occur at very high frequency in the system. So medically, it's a very, very large issue. Certainly a large issue for these children that are born, but even, just dealing with miscarriages and the consequences of these anomalies pre-birth is a big deal. Okay. All right. So what about cytology? Well, if we think about this, in the definition of cytogenetics, we talked about two basic things, right? We talked about a change in the chromosome number. So again, now what we're going to talk about now, from now on, is talking about movements away from the standard set. For us, the standard set is 2N, okay? 2N is our normal diploid number, so we have 46 chromosomes, so we're talking about a variation away from that. That variation can occur either in terms of the number of chromosomes that we have, okay, or in the structure of the chromosomes that we have. These are two very kind of different ideas. So you can either change the number or you can manipulate the physical amount of DNA that you have in a particular chromosome by adding or subtracting or moving it around or something, okay? Now, for the first part of this, we're going to concentrate on the number of chromosomes. And then we'll move on to the later lectures. We'll talk about structure. But for right now, we need to talk about definitions for both of these, okay? All right. Within the number of chromosomes, we have two big terms. One's called euploidy, Euploidy, and the other is called aneuploidy. Okay, and with many things in biology, the little name is the big effect. Big name is the little smaller effect. Okay, and that's the way it works here. Euploidy is this. It's the addition or deletion of entire sets of chromosomes. Okay, so entire sets. So think about it in terms of the haploid set and what you get. For example, you might have monoploidy. So anything that's a euploid will have this suffix ploidy on there. So a monoploidy is a condition where you have basically the haploid set becomes a living individual. Okay. Now some species that's quite normal. In bees and things, you have haploidy diploidy, and many plants have haploidy diploidy, where there's a normal condition. The monoploidy is a condition. But for humans, that would be an aberrancy, something like a non-fertilized egg, a haploid egg, developing into a full adult human. Right. That would be a monoploidy event. Now, there is diploidy or diploidy. Dip is two. We're normally diploid, so it's not an aberrancy for us. This is our normal state, and that's true for many organisms. Almost everything that we've been studying and will study is a diploid. Okay, certainly Mendel's peas and all of that. Now we're going to talk about some variations of that. Uniparental disomy, is going to be there, and all those sorts of things we'll talk about later, which are variants of this. But for us, diploidy is normal. Then there's a whole group that fall under the large category of polyploidy. Polyploidy can include things like triploids, which are 3 times the amount of the haploid set. So it might be, like an egg and 2 sperm or something like that. Okay. There's tetraploidy, there's pentaploidy, there's octaploidy, there's all sorts of things that we'll move up the line on ploidies. Okay, and we'll give some examples of those later lectures. All right. So we've got euploidy. It's this addition or subtraction of entire sets. So you got a complete set that you don't need, or you're missing a complete set. Aneuploidy, as I said, bigger word, but slightly smaller effect. Now, not necessarily a smaller effect in terms of the outcome, but smaller effect in terms of the number of chromosomes involved. So here we're going to add or subtract individual chromosomes, right? So let's pretend that up here somewhere we've got a diploid number, and we just lose one. Everything else has its complement, but we lose one or we gain an extra one or something. That's an aneuploidy event. The suffix that goes here is somy. So we had ploidy when we're talking about euploidy. We have somy when we're talking about just addition or subtraction. A monosomy is that we normally should have 2 chromosomes at one particular pair, and we lose one, right? That's monosomy. We left with mono. Okay. Then there's disomy, we'll talk about later, but trisomy is when we got an extra one. For some reason, we got 3 homologs here. Okay. Then there's tetrasomy, where we've got 4 at one place. Now everything else is normal. Okay. And then there's things like pentasomy and hexasomy and all sorts of things, which are obviously more and more rare as you work through it. Okay. To kind of illustrate this, I just like to use this kind of a little simple example here. So here's a cell. Let's make a haploid number of 3, and we'll just make some little chromosomes. Okay, so these are just the haploid set of 3. You can make them as nice, pretty chromosomes if you want, but here's mine. Okay, so this is chromosome number 1, number 2, number 3. All right, if we take a cell, we make a normal cell that's 2N. That means we duplicate the haploid set. It looks something like this, right? We'd have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3. All right. Okay, so then let's start some aberrancies. So what is this? Okay, well, we have 1, 2, 3, 4, 5 chromosomes. We're supposed to have 6. We're missing 1. We're only missing 1. We're not missing an entire set. So this is some type of somy. So obviously this would be what we would call a monosomy, right? Very straightforward. In this case, 5 chromosomes. In humans, a monosomy would give us 45 total chromosomes, right? Okay, this one is just the complete opposite. We've got normal pair here for the first, normal pair for the second. It looks like we've picked up an extra chromosome, extra homolog down here in set 3. So this is still a somy, right? We haven't duplicated the entire set. So it's 1, 2, 3, or a trisomy. In this case, we've got 7 chromosomes. In humans, we would have 47 chromosomes if we were to do this, right? Then we have this little guy up here. Okay, well now everybody's there. We're not missing any, and we don't just have a kind of 1 extra. We seem to have a whole pile of extra. So you count based on the haploid number. So there's haploid 1, haploid 2, haploid 3, haploid 4. So instead of just having 2 we have 4 of these. So this is some kind of ploidy, and this is classically tetraploidy. Okay, and just one more little example here. Here's this thing. Okay, okay, we've got 1, 2, 3, 4, 5, 6. Well, that's normal, right? We're supposed to have 6 chromosomes. Oh, but wait a minute. This one doesn't have a partner here. It doesn't have a homolog. This one has 3 homologs. This one seems okay. What the heck's going on there? Well, the reason I put this in is to basically remind myself to say this, which is that this is a monosomy of the first as well as a trisomy of the second. What I want to remind myself to say is that when you have one chromosomal aberrancy in the real world, very often you have more than one. Okay, so even though it's a somy, very often when we look at these cells, and there's some very famous encyclopedic view of these things from Boganker. Dr. Boganker was well known for his catalog of chromosomal aberrancies. You'll often see in cells where you end up with a trisomy at one place, there'll be a trisomy somewhere else. Not a triploidy, just a somy. So I just put that in there to kind of think about the real world. Okay, so now we have some names. So let's talk about this change in number of chromosomes that we're dealing with here. Okay, so we're going back to number of chromosomes now, right? And some of the possible causes. And one of the biggies is fertilization errors, right? Fertilization is a big process, whether you're a plant and you're trying to get pollen to exchange DNA insert DNA into an ovum, whether you're a mammal and you've got sperm and you've got egg trying to mate, put together. It's a very complex and difficult process where lots of things can happen, okay? Now, just in the big phase, think about an egg that's haploid. We think about a sperm coming along, it's haploid. If those fuse together properly, you get 2N, you get your normal diploid. But as we know, if particularly if you're springtime and you walk outside and the plants are throwing out pollen, your car in the south at least is covered in sort of yellow pollen from the pine and others. You know that there's plenty of sperm out there, or in that case, plenty of pollen. Well, sperm are the same way, right? When males produce sperm, we produce lots of them. Oogenesis generally only produces 1 egg at a time, so there's an excessive number of sperm to eggs or pollen to ova. So therefore, it's very likely that multiple sperm will hit at the same time, will be in the same place, same time. If that is allowed to happen, then you can end up with a triploid condition, right? And in fact, that's what can happen. Well, think about it though, in the real sense, you've got lots and lots of sperm. In mammalian systems, you've got a number of sperm being reached, maybe hundreds or thousands of sperm at a time. In fact, if you look at an egg fertilization, there's some really nice things on YouTube and other where you can see this. You see these, there are lots of sperm around even one ovum, one egg at a time. Well, this is such a big deal that through evolutionary time, there have been a number of systems developed to try to protect the egg from this multiple fertilization. In mammalian eggs, there's this sort of, the egg just swells almost immediately. It's just an amazing thing. There's known as a fertilization membrane, and it's basically this just almost gelatinous sheath that comes around. It does many things, okay? It slows down some of these sperm from entering. It degrades the sperm themselves, and it protects the eggs and tries to protect from having multiple fertilizations go on. So these things, some of them are just plain not able to get there to the membrane itself, to the outer part of the egg membrane, because they're slowed down and they're digested away. Others can get there, but they simply aren't allowed to function, so that they can't move enough to get the DNA that they carry released. Okay, so it's called a fertilization membrane. I really encourage you to go on the web, just do a search for mammalian egg fertilization and watch the process. I mean, the egg just doubles in size almost instantaneously once the first sperm makes real contact with the system. But you can see where fertilization errors could easily occur. Okay, the other place you see it is errors that occur in mitosis or meiosis, right? One of the biggies, and one that we'll look at a lot, is called nondisjunction. And it can be spelled DYS or DIS, doesn't matter. Nondisjunction. And we'll talk about the process of dual replication as well. Okay. Nondisjunction is exactly what it sounds like. You've got to form these spindle fibers. Okay. For our analogy, you can think of them as big bungee cords. We've all seen what happens to bungee jumpers sometimes when you jump off of something. Some of the cords break, right? Or simply it's a very complex system in here where they have to attach. So maybe you just don't even get proper attachment. So either one can happen. If that happens, you end up with what's known as a non-disjunction event. You pull, instead of separating the dyads into two parts in this case, you don't separate them. You pull the full dyad to one side, you fail to pull anything on that particular chromosome to the other side, and you end up with non-disjunction. Okay. Now this can occur anywhere. It can occur in mitosis. Okay. Although that tends to be short-lived, it only affects the future cells from there. Or it can occur in meiosis, which means it's going to potentially be passed down. This figure, which is very common on the web and other places and in your notes, really need to look at this figure and pay a bit of attention to it. Non-disjunction in meiosis can occur one of two places. It can occur either in meiosis 1 at anaphase 1. In that case, the homologs go together, right? Whatever happens, the homologs go like this, all right? And then once you do that, then you've got this secondary separations that occur. And even if it goes normally through metaphase 2, you end up with these sort of different outcomes. Or you can go ahead and separate and have segregation go on as you normally would in anaphase 1. But when you get to anaphase 2, maybe one of them doesn't separate. This one does, but this one doesn't. Okay, that's what's being illustrated here. That gives you a different outcome. And there are always little questions for on the backs of chapters on chromosome anomalies and things about Billy has color blindness, his dad didn't, his mom did, but blah blah blah. Where did the non-disjunction occur or something, right? He's a thing. There's all these questions. So if you understand where these two things could happen, you can think backwards from the child back here and figure out, okay, did it happen in mom? Did it happen in dad? Did it happen in production of egg or sperm? And then did it happen in meiosis 1 or in meiosis 2? So it's something you should study. Okay? All right, and then we have this concept of dual replication. Now what dual replication really means is that you just basically skip mitosis or meiosis, okay? You go along and the cell is a pretty dumb system, right? The system, it kind of does go in a circle like this. And so you're in G1, you go through replication, you duplicate all those homologs, make sister chromatids. You get up here to through the G2, and for whatever reason, it bypasses mitosis. Now, it can't go backwards. It can't create these chromosomes, these chromatids. It keeps going this way, which means essentially you go into G1 again, which by that point everything's doubled. And to get back to mitosis, it doesn't skip replication. It goes through replication, which means now you got 4 of all these things, and you end up with multiples in these. It's called dual replication. Okay? And it occurs, and we're learning more and more about it because when we go to look at the cell cycle, there are all these checkpoints. There's checkpoints to make sure that the DNA isn't damaged when you go out of G1 into synthesis. There's a checkpoint because all the things that have to go on in replication to make sure that you got it all replicated correctly. And then in G2, before you can go into meiosis or mitosis, there is what's known as an antiphase checkpoint. That antiphase checkpoint has a number of proteins that come into play. They check to make sure that the spindle fibers and the spindle mechanisms can assemble correctly, that everything can happen. If they can't, in a normal cell, what they'll do is they'll kind of slow down the process until the proteins can get there to make the system work. Okay, but they can completely block the system, in which case the cell will eventually keep going and end up back in G1. There's a number of proteins here, and actually one of the primary ones that's here is a ubiquitination protein that ubiquitinates other proteins and activates them, and it's often downregulated in cancers. We don't know whether it's downregulated as a result of the cancer or as leading to cancer, but it downregulates, which means that the system doesn't correct everything and just speeds through this whole process. So it doesn't block it, it actually speeds through the process itself. And one of the cool things about that, cancer is not cool, but understanding the mechanism of this protein is cool. One of the things about what happens is that this protein is epigenetics, not epigenesis, epigenetically modified. So it's actually the DNA is not changed. It's not changed from ATGC or whatever, but the control regions are methylated. We'll talk about epigenesis, and that's really one of the places. But for us, something happens in here. And what happens is we skip over the mitosis and we end up over here in G1 with too many chromosomes. We take ourselves and we go right back through replication, and then we're back up here with too many. Well, you can get a doubling of the chromosome number. Okay. All right, so we've got non-disjunction, which we'll talk about quite a bit. We've got this dual replication, which we'll talk about a lot less, and it's kind of about the basic time we'll actually mention it. Now, after now, I'm going to start moving into some of the next lectures will be about some of these actual anomalies and how they occur. First, we'll talk about changes in chromosome number anomalies, and then later lectures we'll talk about actual structural anomalies.